This tutorial will demonstrate PCR, restriction enzyme digestion, and DNA electrophoresis using controls of the cystic fibrosis case as an example. The first thing to do is to click the DNA button on the silver button bar, and you will see the inside of the CASEIT folder. Now I'm running this on a Mac. If you are using a Mac, avoid these two buttons. The reason for that is this is a Windows application running on a Mac, and I'm going to very carefully show that this is the Windows system inside the Mac. If you start clicking in there, it can get confusing. So bottom line is just avoid these two. So to continue, I'm going to double click and open the cases folder. And the cystic fibrosis case is inside the genetic disease cases folder. Here it is. I'm double clicking. And case A includes the controls. Case B and case C also include the same controls, but I'll just use case A. Use the Brown family inside case A. So here are the three controls. I'm going to avoid clicking any of the family members because I don't want to run that case. All I want to do is demonstrate features of the software. So I'm clicking on the first control, shift clicking to highlight them all, and then clicking the open button. Before running PCR, we need forward and reverse primers. So I will go to the primer button and select that file. A quick way to run PCR is to shift click to highlight the three controls and then go to the Run button and run PCR. And then the PCR fragments appear here in the Open in Process window, also indicated by PCR in parentheses as a suffix, and the prefix are these three arrows. So anytime you see that, those are PCR fragments. The next thing we want to do is to digest those fragments with an enzyme. So I will go to the enzyme button on the silver button bar, and I'll open up the enzyme MSP1. You can also do that through this colored button. And here is MSP1 showing that it's a stagger cut, and it shows the nucleotides associated with that cut site. But we don't have to do that because that enzyme was inside the case A folder. To digest those three PCR products, we'll shift click to highlight them. And this enzyme is active. We can see it up here. I'll use the Cut DNA button and select with a single enzyme. There are two different ways of loading these and running the gel, and I'll demonstrate both. The first method would be to select them individually, click this Load button, and then the information for the loaded well appears here. Click the second one and load it, and you can see that information is now also in the loaded window. And then you can click the third one and load it, and then you can run the gel and stain it by clicking the purple button. There is a faster way of loading wells and running gels, so I'll go through that procedure now. Go up to the Clear menu, but select Other Options, and for 8-well gel, clear wells, lanes, and search results, but don't clear the Open in Process files. Then what I'm going to do is select those three digested PCR products. This time we'll use the Quick Load Run button and go to DNA Gel, load wells and run gel in one step, and stain DNA fragments purple. There are two other videos in this series that describe DNA digestion and electrophoresis in more detail, parts 1 and 2. In this video, I'll demonstrate how to visualize forward and reverse primer sites on any DNA sequence, and also how to animate the first step of the PCR process. To do that, I'm going to first activate the primers to see those forward and reverse sites. And the question is, where are they located on the DNA sequence? I'll just arbitrarily select the homozygous normal one. So we've selected the DNA sequence. You can see the information here. I'll go to the DNA menu and show DNA base pairs and PCR primer sites for the open file. When you do that, the sequence appears in this upper window. And again, here are the forward and reverse primer sites, but what does that really mean in terms of a double-stranded DNA molecule? Here you can see the double strand. When you load files into CASEIT, you're only seeing one of the two strands. Here is the double-stranded configuration for the first 60 base pairs of that sequence, and you can see that it matches these 60 base pairs here. To see the forward primer site, I'm going to click this button. Here it is. And to see the reverse primer site, I'll click this button. Here it is, and it's highlighted here. Click the forward primer site. Here it's highlighted. Now notice it says GCAACAATTC. That corresponds to this. But in reality, the forward primer is going to anneal to the complementary strand of the DNA molecule. So this is what it's annealing it to. And notice these bases are written upside down and backwards. Again, I'll click the reverse primer site, and here it shows the GCTTT, GTTC, which is right here, because this reverse primer is going to anneal to this complementary strand. If you want to see both sites, there they are, separated by a whole series of bases that can't be shown here. So that's why this symbol represents those bases that would be in between. And if you wanted to animate this to see the very first step of the PCR process, you'd click the animate button. So that's the first step of the PCR process. Then there'd be many, many other steps to the process that we can't show here. You can click the Go Back button and then show both sites again. For this particular example, there are several hundred base pairs between the two primer sites. And to demonstrate that, here's the forward primer site and the reverse primer site. I'll do that one more time. Forward primer site, notice where it's located. And then if you watch that and click the reverse primer site, these are the bases that are in between those two sites. So you would not expect those PCR products to be very large. And in fact, if you took a look at them, you can see there are only 383 base pairs. And then to see their actual sequence, there it is. So those are the fragments then that are going to be digested by the enzyme MSP1. And you can do that by activating MSP1. And then you can cut it with a single enzyme, and then those are the sequences and the sizes 215 and 168. Again, the sequences are those four, and the sizes 215 and 168. You can see that on the gel. Go to the gel blot button and go to run, our run. And then if you click in the lane that had the homozygous normal fragments, which was lane 3, and then when you have to click off of the fragments and then hold your cursor over it, that's 168 and 215. And of course there are two 215 fragments on top of each other and two 168 fragments on top of each other, so it looks like just two bands, but for the purpose of the simulation, there's 4 fragments. For real PCR, there would be many fragments stacked up on top of each other. Migration distances for lane 3 are shown here. I just clicked on that lane, and then you can see lane 2, lane 1. But going to lane 3, you can see the migration distances from 0 to 100. To get back to those sizes, we just select band sizes, show band size for lane 3, and then it shows those sizes that we talked about before, but they're in kilobases rather than bases the way they were down here. And also if you click here, again 168, that's rounded to 0.17 kilobases, and 215 bases is rounded to 0.21 kilobases. This concludes the tutorial for PCR, restriction enzyme digestion, and DNA electrophoresis.